Hi everyone! So now we're going to go ahead and go through mitosis, get into some of those nitty-gritty details that we didn't in the cell cycle video. Okay, so there's 5 phases of mitosis, and I'm going to move this over. You'll see that I kind of have stuff underlined as a hopefully helpful way to help you remember what's happening and everything. So prophase is the packing of chromosomes. So P and P. Pro-metaphase is purging the nuclear membrane. This just means we're getting rid of the nuclear membrane. I could not find another good P word. So if you have one that means get rid of or dissolve, let me know. But what I got for y'all is purge. So P for pro, M for the metaphase, pro-metaphase, get rid of the nuclear membrane, purge it. And then metaphase, all the chromosomes are in the middle. Anaphase, the chromosomes are pulled apart. And telophase, 2 nuclei are formed. And I do have a little hand motion trick for you guys to remember all of these. It does not include prometaphase, it's just prophase, metaphase, anaphase, telophase, but you should be able to remember that prometaphase is between prophase and metaphase just because of what the actual word looks like. So here we go. Prophase is packing of the chromosomes. So you can see that my fingers are interlaced and everything's packed tightly. Metaphase, the chromosomes are in the middle. Anaphase, they're pulled apart. Telophase, 2 nuclei. So there's 2 circles and they're on my eyeballs, 2 nuclei. So if that helps you, great. If you knew these stages already, also great. But there you go. If you want those hand motions, feel free to use them. So now we're going to go ahead and look at each of these a little bit more closely. So I'm going to move my video again, starting with prophase. The chromosomes are going to be condensing or packing together, which just means everything is getting really close so they're smaller. If you think about it, if you're trying to pull things apart and do stuff with something, you would prefer that it's pretty compact because it's just easier to deal with. So you can think about like a super stretched out slinky. If you were trying to do something with this huge, really long slinky, it would be kind of hard to drag it around everywhere. And so you'd probably want to push it together and then you would carry it to wherever it needs to go. So that's what your cell is doing here. Your chromosome or your DNA is like all messed up and everywhere, and it's going to compact it really, really tight into our chromosomes that you can see in this little picture right here. Now they're compact, they're small, and it's much easier to pull them apart and do whatever we need to do with them. Another really important part of prophase is these little yellow things over here. So you'll see there's these 2 yellow like log things in the middle. Those are centrioles. And the centrosome is where the centrioles live. So the centrosome is not drawn here, but technically you can kind of think about it as like a circle around these centrioles. And there's 2. Eventually that one is going to go to this side and the other is going to go to the other side. So those are centrioles, and centrioles are really important because they're going to make your spindle microtubules. And remember, your spindle microtubules are your arm that reach out to your kinetochore. Your kinetochore is attached to the centromere of your chromosome, and they'll help with pulling everything apart in anaphase. So the reason that one has to go to this side and one goes to the other side is because we want to approach the chromosome from both sides. That way we can pull to one side of the cell, and that makes splitting the entire cell in half easier. So the next phase is prometaphase. And in this phase, the nuclear membrane is dissolved. And a good study technique whenever you're studying is to say why. So not just knowing, okay, prometaphase, the nuclear membrane is dissolved. But if you really want to remember it and get it in your head, it's really good to always think, but why does this have to happen? Why is this important? Because that'll give you a deeper understanding of the material, and it'll mean that you can remember it better and recall it later on your exam, on the final, and future classes, and if you're taking the MCAT, on the MCAT. So let's think about why, and I'm going to go back to my previous picture in prophase. You'll notice here that we have a nuclear membrane with our chromosomes in the middle, and our centrioles that are making those microtubules are all on the outside. So the microtubules can't get to the chromosomes right now because they're protected in the nuclear membrane, and the microtubule can't get through the nuclear membrane. So in order for anything to happen, we need this nuclear membrane to disappear because our centrioles and our microtubules are on the outside. So if we ever want to go to metaphase or anaphase, we need that nuclear membrane to dissolve. So that's what's happening here. The membrane disappears, and now the microtubules can come and kind of start attaching and trying to get to metaphase. And so remember, your microtubules are going to connect to your kinetochore. Your kinetochore is at the centromere of your chromosome. The way that I like to think about it is that your chromosome is something that the kinetochore is holding onto. So the kinetochore is a hand holding onto the centromere, and the arm is the microtubule that'll help pull everything apart. All right, let's go ahead and go to metaphase. So metaphase is pretty simple. Everything is in the middle. So a microtubule is on each side. So looking at this picture right here, microtubule, microtubule on each side, and they're both going to be attached at that centromere using the kinetochore. And they're both going to be pulling a little bit, so there's a little bit of tension to keep everything in the middle. So you can kind of think about it as an equal tug of war. If this is the rope, and you know how there's always like a little yellow flag in the middle or something, think about the chromosome as that little yellow flag and the rope, and each side is pulling, but nothing is moving. It's just all staying in the center because it's equal. So then anaphase, imagine that somehow they pulled way too hard, both sides of the tug of rope, and the rope just completely split in half right down the center of our little yellow flag. So now we have a little bit of our yellow flag or our chromosome going to each side. So here we went from sister chromatids, where here we have 1, 2, 3, 4 chromosomes, because when we count chromosomes, we just count the number of centromeres. In anaphase, we have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. So this is important with DNA molecule and chromosome counting, so keep that in mind whenever you watch that video. So yeah, anaphase, the tug of war rope broke, and at this stage, our sister chromatids, remember they used to be identical twins, and they were just one chromosome, and now they're apart, because remember, if twins aren't next to each other, we don't call them the twins, we call them by their name. So at this point, we no longer have sister chromatids, everything is now its own chromosome. All right, telophase. So telophase is pretty much just the opposite of prometaphase and prophase. Prophase and prometaphase are all because we want to get everything pulled apart. So it's basically preparing for anaphase. And after anaphase happens, we want to get back into the interphase of the cell cycle, go back into G1, which means that we want everything relaxed again. We want a nuclear membrane because we don't want to just allow anything to get to our DNA because that would be bad. So telophase is undoing everything that prometaphase and prophase did. So this means the chromosomes are relaxing. Think about stretching your slinky out. The reason we want the chromosomes relaxed is because whenever they relax, it's a lot easier for different enzymes to go up to the DNA, do transcription and that kind of stuff. When it's really packed tightly together, it's really hard for them to access the DNA. And we know in interphase, we're doing cell growth as well as just normal regular activities. So we want to make sure all the enzymes can access what they need to. We also reform the nuclear membrane. Like I said, we don't want those random toxins just affecting our nucleus and getting in and destroying our DNA. So we need that little protective barrier. And you'll notice in this phase that we have 2 nuclei. Here's your first nuclei, and here's your second one. So cytokinesis is kind of also seen in this picture. You have this is called the cleavage furrow, where the cytoplasm kind of goes together. And that's because it happens pretty much the same time as telophase. So basically all we're doing is chopping the cell in half, give each cell 1 nucleus, half the cytoplasm, and half the organelles to keep everything fair. So remember with mitosis, your genetic results is that you have 2 identical cells, and they are also, the 2 daughter cells are identical to the parent cell. And cytokinesis involves the cytoplasm. They both have cyto at the beginning, so that should help you remember that. All right, and that is the end of the mitosis tricky topic video.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.